Welcome to Under the Microscope podcast, where we spotlight materials and nanoscience. Under the Microscope is the flagship podcast of the Science Talk, and here we explore the fascinating world of materials and nanoscience. Our goal is to provide a stage where scientists can communicate their work and interact with the public. With that in mind, with every episode, we introduced you to a scientist working in the field of materials and nanoscience. Join us as we delve into the groundbreaking research, innovative discoveries, and the inspiring stories of scientists making waves in this field. Welcome to the Science Talks podcast. I'm your host, Pranoti Kshirsagar, and today we have with us Krist Gernaey, who is a professor at the Technical University of Denmark, close to Copenhagen. Hi, Krist. How are you?
Thank you. I'm fine.
To begin with, what is your first memory of being fascinated with science?
Yeah, I think that I have to go back to university times when I was studying at Ghent University. And I discovered that some of the professors that were teaching classes, they were very enthusiastic about the subject of research or the topic they were teaching about, which made me ask questions. And I think that's also resulted in starting a master thesis with a specific supervisor. Enthusiastic about this field, you could only get caught by his enthusiasm.
It was quite infectious, their enthusiasm.
Yeah.
And now that you are a professor yourself, do you understand it now from the professor's perspective? You are in their shoes now.
Yeah, definitely. I mean, it's fun to see how people react when you talk with enthusiasm about your topic. And it's also always fun to see how students get engaged in discussion and in the end maybe decide to go for a master thesis, go for a PhD project. There's so many different topics. And I always feel very happy if somebody chooses now to go in the direction where we now do our research.
I can understand. And speaking of that, so can you please explain to me your current research in super simple words? Okay, just like super simple. What is your current research? What are you doing?
So the core subject, what we call industrial fermentation technology, which means that we are essentially looking at everything which is around the cell. We are not looking what is inside the cell. We try to look at what is outside. So that means we look at bioreactors, we look at separation processes, we look at mixing, transferring bioreactors, we look at scaling up, scaling down, and measurement devices to be able to measure in real time what is going on in this bioreactor.
What do you mean by industrial fermentation? Give me an example in terms of which industries are we talking about here? I'm pretty sure we are not talking about the sourdough bread, because that is what comes to my mind when I think about fermentation.
If we talk about industrial fermentation, we talk about a process that runs in a large bioreactor. So we talk about a process that runs at 20 cubic meters, 100 cubic meters, 200 cubic meter scale. We typically work with is, for example, enzyme production, could be production of bioethanol, and it could also be a protein, this type of production processes.
Which is produced on an industrial, like a large scale, essentially.
Yeah, the products we are involved with, it only makes sense to produce them if you can produce them at a large scale. Well, for example, if you have enzymes which are used in detergents, it doesn't make a difference if you can produce 1 kilo, you need to produce many hundreds or thousands of kilos to be able to make a business. So the dream is that you can do an experiment in the lab at very small scale, get an idea about how your organism behaves, to get an idea about kinetics and which factors play a role in trying to make your organism feel happy and produce, then use models to predict how will this same organism behave if we now try to imagine that it's operating at large scale.
So the difference between fermentation and a chemical reaction, like the classical chemical reactions, is that in fermentation, the process is catalyzed or activated or is done by microorganisms. And in a chemical process, you add different chemicals and then the reaction happens on an atomic or a molecular level.
We have a microorganism, which could be bacteria, could be yeast, could be fungi, but organism is key to transforming some raw materials to a product. Of course, another challenge is when you then have produced your product, how can you isolate it and actually bring it to a concentration, to a purity?
And the products from your processes, the different processes that you work on, these would act as the raw material for the different industries, be it consumer goods, be it like shampoo or detergent, or it could also be, what else could it be?
Yeah, for example, we've been involved in some project where we try to produce biofuels. So ethanol, also been involved in trying to produce other alternatives like butanol. We have done quite some research related to enzyme production. They are also produced at large scale produce a sufficient amount. It can be used, for example, as an ingredient for food production or as a technical enzyme. We need to have decent quantity of enzyme. That's why we do these bioreactors. You do typically experiments at a very small scale, which we call screening. And then in the small scale, you isolate some candidate strains which are producing well. Then you try to increase the scale to see if they keep on producing well, for example, in a lab scale bioreactors, or bench scale, and then finally at production scale. One of the most fascinating things I think in my work is to see this interaction between, on the one hand, scientists which try to improve the microorganism, which can, of course, productivity, product concentration. And on the other hand, there is another group of scientists which try to work with process optimization, where you try to, for example, change feeding strategies to also increase productivity. These have to work together. These two, let's say, branches of science have to work together to be able to get the best out of such a fermentation process. I think is a fascinating aspect. And then also the interaction between what we call upstream, so the process itself, and then downstream, where you try to isolate from the fermentation.
To take it, like isolating, meaning that take it out now to be able to use it. And okay. You said bioreactors. You also mentioned biofuels. In this case, in this particular case, when you say bioreactors, you are referring to the microorganisms, right? Like be it bacteria, be it yeast, be it whatsoever. That is the bio component of this bioreactor, right?
The start of everything we do or we are involved in is typically that there is a conversion that is catalyzed by a microorganism.
And how do you make these microorganisms? How do you do that? Because this is like a completely different field for me. This is like completely different because when I did my PhD, I was making devices, medical devices for testing in vitro with the cardiomyocytes or neurons or retina cells and so on and so forth. But there, I know the process. Where do the cardiomyocytes, which are the heart cells, are coming from to be able to do the test in the lab? So in your case, the microorganisms that you have, do you like, how does that work? Is there a farm? Do you buy them? Do you grow them? You can't grow, like, how does that work?
Well, it's both. So on the one hand, you can buy strains in culture collections, and these strains typically they are very well characterized, and it's known that they can produce, for example, a specific product. They are very well characterized, so these you typically would buy. If we just need a simple process, for example, test a new device in a bioreactor, then maybe we would just buy some baker's yeast and then just run a fermentation with that. And there are the genetically modified organisms, so GMOs, and the scientists have managed to change the DNA of the organism such that it can produce either a specific product or it can produce more of a product which it already produced naturally. So you can build in the capacity to produce a new product, because many companies, they will work with what is called a production platform, which they know how it behaves more or less. Then they will try to extend the capacity of this production platform to produce, for example, new molecules. Alternatively, you can also have a natural producer of a specific product, which you try to modify to be able to produce more of it. Seeking good fermentation is often that you need to produce a product that can be present in the fermentation broth at a very high concentration. The higher the concentration of your product in the fermentation broth, the lower downstream processing cost will be. So that the lower the cost will be to isolate product from the fermentation.
You are opening a completely new dimension for me, because as a material scientist, I know the 2D materials, the nanomaterials, the colloids to some extent. I know a little bit about the biological sort of systems. But this is like the microorganisms. This is like a completely new field for me, but it's so cool. And also, what is really, really interesting for me is that there are several different conventional disciplines of science coming together to make this happen. From lab to the production scale, you also probably need the modeling side, like the theoretical knowledge as well. How did this come about? Can you tell me about your career journey to where you are today? So a professor at the Technical University of Denmark. I'm pretty sure the 5-year-old or a 10-year-old Chris wasn't thinking, okay, I'm going to work in the fermentation and industrial fermentation. Like, can you tell me about your career journey so far? How did this happen? Because your work is really fascinating.
Yeah, I think so. I've grown up around Ghent in Belgium, and I think when I went to what you would in the States call high school, then I started getting quite interested in the more scientific topics which we got as subjects. So, for example, chemistry, especially organic chemistry. We didn't get so much about fermentation, but it was quite clear that I wanted to study something at university which was related to natural sciences. And in the end, I embarked on a 5-year education at Ghent University, which was located at the old agricultural faculty, which had been converted into a faculty, I think it was called Applied Logical Sciences. So basically, the topics that they were used for as research topics there was, on the one hand, food science, but on the other hand, also environmental microbiology. And I think the education that I got was also very broad, and it actually resulted in the fact that I developed also very broad interests in different subjects. For my master thesis, I was inspired by my supervisor, which is called Willy Verstraeten. And he was working at this Center for Microbial Ecology. So they were trying to use microbes for environmental purposes, trying to produce methane from waste products, or tried to do environmental remediation of soils. I embarked on a master project there, which was focused on anaerobic digestion, process where you convert organic material to biogas using a mixed culture. And it's quite a slow process. So the experiments we were doing for my master project, they lasted literally 30 days, 60 days, whatever. The thing I learned is that I was not patient enough for that type of work. So when I then decided to go for a PhD, I switched to aerobic processes, which are faster. I also started developing an interest for measurement, characterization of systems, also some modeling. So I did development of devices which could be used to measure, to quantify activities of specific groups of microorganisms.
And this was also at Ghent then?
Like,
yeah. Okay, sorry, go on.
It was called Center for Microbial Ecology at that time.
So you changed departments, because I'm just thinking the evolution and the change of the names of the departments or the director. It was originally agriculture, then it was
the faculty was the former faculty of agriculture engineering, and it worked at that time already in the faculty of applied biological sciences. So
and then you switched for your PhD.
You could still study there how to dismantle a tractor or how to build a stable, but there was also a lot of focus on food science and so on. So it was a traditional agricultural sciences, also agricultural related economy and so on. And the other hand, all these more new things like fermentation, ecology, and so on and so on. I did my master thesis and my PhD thesis at the same lab, so the same supervisor, Willy Verstraeten. And he was always very enthusiastic about these topics. And that also meant I spent quite a lot of time in the lab. I spent so much time in the lab that at the end of my PhD, I decided that I wanted to focus more on mathematical modeling. So for my postdoc project, I started working on mathematical modeling. I've been doing that from, I would say, 1998 until about 2004, 2005. I had postdocs at different locations. I had a 2-year postdoc project at Ghent University, École Polytechnique de Montréal, where I had a postdoc grant working on a similar topic. And I came to DTU, where I was postdoc for 3 years, or as it was called, assistant professor at that time. And I went to Lund University for the last postdoc, where I also worked 1.5 years on a modeling project. I returned to the University of Denmark in 2005 as associate professor, and then I started teaching and having PhD students. Also decided to change direction, so I'm still today working on modeling of wastewater treatment systems. Since 2005, there has always also been a strong focus on trying to develop activities related to fermentation processes. I've been starting different PhD projects in this period from 2005 until 2012. And in January 2013, I got a full professorship in industrial fermentation technology, which has been a very exciting period. Many things happened. I have worked with many brilliant people, PhD students, postdocs, master students, many different companies. 2014, I also became the head of our research center, which is called Process and Systems Engineering Center. And that is also something I enjoy a lot, because what we try to do is to have an approach which is a combination of experimental work on the one hand. So we focus on fermentation. I also have colleagues that work with biocatalysis and with dowsing experimentally. On the other hand, we try to work with mathematical modeling approaches to be able to take these experimental data, model them, try to use the models to convert this to, for example, a food process, and try to predict how well it behaves. It has also been extended with techniques like life cycle analysis or techno-economic analysis of different process alternatives. At some point, I also had a bigger project where we made some startups. Now I also started a company together with some other people, totally focused on trying to establish more circular processes. So try to work on reuse of, for example, processed water or reuse of organic solvents in industry. CEO of the company for 4 years, then somebody else took over, and I returned to being a full-time university professor. So I've tried a little bit of everything.
It's quite interesting that during your master and your PhD, also you did a lot of like experimental work, and then for postdoc you switched to modeling, more theoretical work. Was it because it was too much work, like the lab was too dirty, or it was like too long the process, or and you? What was the reason for that?
I don't think the lab was too dirty. That's certainly not the case. But on the one hand, I've done a lot of lab work. Also, for actually during my first postdoc, the first 1.5 years, we still did a lot of lab work. Maybe we have been so much in the lab that we got a bit tired of it. Then my first postdoc was also in a group at Kent University still, but which was entirely focused almost on mathematical modeling. So the experimental activities were a minor part of what the whole group was doing. Basically, this also created an interest in trying to work more on mathematical modeling approaches. So I've worked at, especially here at DTU, also when I worked at Lund University, I had projects which were entirely focused on collecting data outside the university, try to apply mathematical models to these data sets, work more with theoretical model development, and from how it started, mainly lab work. I didn't know when I started my PhD that it would end like this, of course not, but it just stirred an interest, and then you try to follow it. And I'm glad I've done this because I learned some new things. I also have decided to stay in the lab, and I don't say that is bad, but I think I have a broader horizon now by changing direction from time to time.
Yeah, absolutely. From the location perspective, you were in Belgium, you were in Canada, you were in Denmark in a bunch of labs in Denmark. So again, DTU, then Lund, then back to DTU now. So this is quite interesting. And now you're a full professor at the DTU, right? And the head of the Priosis department.
We have a lot of departments. So I work at the Department of Chemical and Biochemical Engineering. By the way, until 2007, it was called Department of Chemical Engineering. But then they realized that there's so much focus on biological, bio-based activities that the name of the department was changed, and also includes biochemical aspects of the biotechnology. Yeah.
Wow, that's quite interesting, and it is also interesting because in terms of like the moving around for professional, for the career part, it is great to move around, like get from lab to lab, but that is also the time when your personal life or private life is also growing, right? So how did you manage the work-life balance?
It's a difficult subject because I mean one of the big challenges I think for any scientific career is that at the time where you're supposed to establish a family, you're also exactly in that period of time where you're supposed to spend a lot of time on work. At the time when we got our first child, that was also the time when I had to start teaching at DTU. And normally, of course, the first time is very intensive. I remember that some weekends our son was ill, and then at the same time I had to prepare teaching for Monday, and that was not always a very good combination. It's also difficult. I mean, in the end, we ended up having 3 children. And at the university, it has been like this that every year I had the feeling I just got a little bit more busy. The thing that I've learned is not to worry too much about things. So I can work until I sit at my computer and then I put my head down and I sleep immediately. I never think about my work when I lay in my bed. And then a happy life. At least you get your sleep and then you can be around for the family when it's really required. But it's not easy. And I know my wife has often taken much more of the load at home compared to me, and it's not something you are proud of. It is actually a difficult situation.
Thank you for sharing that. It's definitely hard, should be spoken about because your personal life, putting your roots down, expanding your family often collides with also very, very busy times in your career as a scientist.
My oldest son, he's now trying to find out what to study himself, but he stated a couple of years ago that he thought he would never work at university because he had seen at home what it takes. It is really a challenge to keep this work-life balance.
Yeah, definitely. Definitely. You also mentioned that you have a startup, which you were the CEO of for like 4 years and then you went back. What is it called?
It's called bioscavenge.
Bioscavenge. Okay.
Bioscavenge comes from the fact that we try to scavenge things from a process, typically from a bio-based process. I can also say it's not easy to start a company, especially in this area, because it's fine to do things in the lab and it's fine to do pilot scale. but it's very difficult to end up in a situation where things are actually implemented at large scale, because then you need to involve engineering companies, and everybody has a different idea about how things should be done. Why any client has different standards on how things to be done, and this becomes very complicated. It has until now for sure been a very enriching experience. I mean, we came in contact with some companies, some potential customers, which we never would have talked to if we hadn't made the step to make this startup.
So, Chris, it's very clear to me that you love the research and the science and like different aspects of research and collaboration and so on and so forth. Can you please pick one research project that you're most proud of and explain it to us in simple words in the section we call In Other Words?
Yeah, at the moment, I actually wanted to mention two things, but there's one project we worked on for 8 years, which was a larger project, which was focused industry academia collaboration, which was called BioPro. And in the frame of this project, we managed to transfer quite some things which we normally thought were more university things, so more theoretical. We managed to demonstrate them at pilot scale or full scale. And resulted in the creation of, I think, close to 10 different startups that try to bring this to also to commercialize. Then what is really interesting at the moment, I think, is that we work on a new startup idea, which is trying to produce the chemical sensors to measure glucose, lactate, ammonium in a fermentation process. And the new thing about it is that until now, if you want to do these measurements electrochemically, then you had to immobilize an enzyme. For example, in the case of glucose, we immobilize glucose oxidase, which in the presence of oxygen can convert glucose, and it will generate electrons, which you can measure. So the amount of electrons is proportional to the concentration. We managed to establish chemical sensor which doesn't need the enzyme anymore. So instead, some nanomaterials which interact with the glucose also generate electrons and give a clean signal. Very promising, because enzymes, when you immobilize them, you get problems, for example, thermal stability and so on and so on. And these can avoid if we no longer need an enzyme to do this type of measurements. Of course, we focus on fermentation, on bio-based production. This could potentially also have applications into, for example, the biomedical world.
So the BioPro project that you mentioned that went on for 7 years, which also led to 10 different startups. So Bioscavenge was one of them. And the second one that you mentioned with the sensing, the biosensing, this was also from the BioPro project?
No, that's a completely new development, which has only taken the DTU, and which gave me the chance to work with DTU's innovation environment, which is called Skylab.
Oh, okay.
The past 10 years, DTU has put a lot of effort in creating an environment where students, but also faculty members can actually develop startups. So there's quite a lot of support compared to the time where we established Bioscavenge, and now it's quite different. So, for example, applying for soft funding, getting also learning how to pitch projects, and so on and so on. There's also a mentor arrangement, which I think is very useful. We have a couple of experts from industry, a lot of experience, and they give feedback on the developments that we present to them and on the street that we think we should follow, and also quite a different opinion about things. And that's very nice.
Okay. So this would be your second startup, the sensing one.
Yeah, the second one that I would be involved in starting up from scratch.
Okay, that is quite interesting. Yeah.
I've also been involved in some other startups as an advisor that I'm on now. Also, the BioPro project, we, for example, had another startup which worked on a different way to collect measurement data in a bioreactor. And I've been an advisor for that company for some years. Was down because of lack of interest from industry to actually purchase technology. So
Okay.
you have to try to develop your ideas, and that was very important.
Yeah, absolutely. Absolutely. The funding agencies who supported your research, of course, but also your startup.
Funding, I've been very lucky because, on the one hand, my professorship is sponsored by a company. The nice thing about this is that I have some basic funding, which is supplied by this company. So they pay an amount of money to DTU every year, and I can use this to start, for example, new PhD projects. The nice thing also is that I have this collaboration with a company which, at the time I got my professorship, was called Enzymes. Since the merger with Christian Hansen, it's now called Novonesis. But there's nothing that can stop me from also doing projects with other companies. So I've been working with different companies since I got my professorship. And that's one of the nice aspects of being out here. I think there's also a very specific thing in Denmark, which is that we have foundations. They also sponsor research. Basically, we can apply for research grants with the foundations. If you're lucky to get grants from, for example, the Novo Nordisk Foundation, which opened up for some completely new possibilities. People I work with have also applied for grants from the Carlsberg Foundation, have got money from the Willem Foundation. And then, of course, We also have grants from, let's say, governmental funding. So we got several grants from an organization called the Innovation Fund Denmark, which is funding research, I would say, that is supposed to result in application in a process or a product. We've also received money from the Free Research Council. So the Free Research Council, they, let's say, support research, which is more, maybe a bit more basic compared to Innovation Fund Denmark. It allows us to have a PhD project to spend on a topic which is maybe new for us, goes in a new direction. That's also very nice. And we've also been very lucky once in a while to get some funding from the European community, so EU money. Of course, that also means that getting money from different sources, that brings us also to one of the negative aspects of maybe working in this scientific world. So quite a lot of time on applying for money. Too much with the field to be able to keep a group of, say, 15, 20 people that I supervise or that I work with to keep them happy, to supply them with funding so that they can have a salary.
So, if you had 3 wishes to improve your research experience, this would probably be one on the top of the list, right? Like, if you had 3 wishes to improve your research experience, if I were to ask you that, what would you ask for without me promising anything to you?
One of the things I would have wished is that I could have spent a little bit less time on applying for money. We spent quite some time on that. What is also difficult, I think, is that since we have several sources of funding, the way we have to do the administration of the different projects is also different. A bit complicated to do project administration. And any funding body, any donor of a grant has some specific rules or issues which we have to live up to. That sometimes makes it a bit more complicated. I should not complain. I think I've been lucky that in many cases we applied for money and we actually got a grant. Of course, I've also experienced many times that we got a no. And that is also one of the big challenges, I think, nowadays in research. Young people, when to start research careers, I mean, you have to start somewhere. So at some point, you start applying for money. And at the start, when you get a no and then a no and a no again, it's very difficult to keep the motivation to keep on continuing. The older professors, the more experienced people, they have a task. They should try to help the younger scientists to be able to establish themselves. It should not be about me, but it should be about us.
Yeah, definitely. So that would be your first wish then, to have a funding system, the way the research is funded, more optimized in a sense, so that the scientists can actually focus on the science and the research rather than the frills around it.
I don't say that all funding should just be given to people like that. It could be nice if you had some kind of basic funding that allowed you to, for example, have 1 or 2 PhD projects where you establish yourself, and then at the same time maybe you could apply for some extra grants. Yes, fine, but at least you could start something.
The basis is covered at all times.
Because the start of research here is also you have to start teaching, and typically people also often have children at home, and this is not an easy combination.
That's a very good point. So that would be your first wish. You have 2 more wishes, Krist. If you have 2 more wishes to improve your research experience.
Yeah, I think one of the wishes is that I think I should have started focusing on fermentation a bit earlier than I did now. So
Why do you say that?
yeah, it's because it's such a rich field that there's so many different things you can do. So that could have of course have been interesting to start earlier working on fermentation, to change directions. And I'm also very grateful that I got this chance. At some point, people at DTU decided that they wanted to hire this weird guy and start activities in this area. And that thing has been a very good decision, I hope.
Yeah, definitely.
Good and very interesting to be allowed to do this.
Yeah.
It's sometimes difficult to explain how I combine these different things, but in the end, when we work with mathematical models, we often say that all the systems we work with, or most of them, they are non-linear. So the type of equations we use are quite simple. Talk about a pharmaceutical production process or a fermentation process or a wastewater treatment plant. We use same modeling techniques on these different systems.
Absolutely. So that would be your second wish, to start working on fermentation earlier than you did in your career. So, and the third and the last wish, what would you ask for?
I think then I have to think back to when we announced that now we had a third child on the way. Then one of my brothers wrote to me, "Hey, now you need 30 hours today." It would be nice to have days which are 30 hours, because there's so many things that I would like to do, and you always have to stop at some point and say, okay, but now we also need to sleep. I think I'm happy that I can work on this, the place where I am and on the topics I work with. It's difficult to say no, and that's maybe one of my weak points. It's very difficult to say no. There's always new, interesting developments which you would like to be involved.
But if it's a wish, why stop at 30 hours? Why not go for 60 hours or 96 or 100 hours?
Yeah, but then I would have to move to another planet, I think. And as far as I know, this morning it was still like this that my family would like to keep me close by. So,
But it's a wish, right? I mean, everything else would align accordingly. But yeah, even with your wishes, you are a bit more realistic, probably. But
also, we can make this wish because basically we are very lucky because we can work on something which we really like, and then it's not so difficult to spend a lot of time on this.
yeah, absolutely.
It can be very frustrating for the people we live together with that we like this topic so much.
Yeah, I can imagine. But it's I mean, it is interesting. The research is interesting, the science is interesting, and combining different expertise and making a difference and discovering new things. I mean, I find it really interesting. I mean, I created a whole podcast just to be able to talk about science. So I definitely think it's interesting. Chris, this has been wonderful. But before I let you go, I have to ask you one last question, and that is about your time on the Blue Sky account of the Science Talk. For those who are not familiar with the concept, Krist will also be taking over a Blue Sky account of the Science Talk for one entire week from Monday to Sunday. And he will be posting from the Blue Sky account as himself. So this is the curation concept that we have, which is in addition to the podcast, the video podcast, if you're watching on YouTube. So in the week that you're taking over the account, Krist, what can the followers expect? What can we look forward to?
I think provide some insight on how my life is as a scientist. So no 2 days which are the same. I meet a lot of interesting people. I come different places, and I think I will try to document some of that. Hope I can make it interesting. It can also be that people, when they see a couple of posts, they start thinking, gosh, this man should be arrested. But I mean, that's also why I like my job a lot. I don't have 2 days which are exactly the same. There's always something new popping up. It can be a new problem that we have to solve. It can be a new collaboration partner that suddenly pops up, or a new student that is coming by to ask questions about a potential project. So there's always something new happening. And also one of the things I like a lot about my job is that I meet a lot of different people. It can be students, it can be people from companies, it can be other professors, but that is also one of the very interesting aspects, I think, of what I'm involved in.
And we will get to see all of that from you when you're posting on the account. So your life as a professor at the Technical University of Denmark, the DTU, how is that going to be like? So the campus, what you do in a day, in a week, your research, everything. And it is definitely interesting, your life as a scientist. And we are definitely looking forward to your posts on the Blue Sky account.
I hope so, because my children sometimes say that I'm a very boring professor. So,
well, they should listen to the podcast and watch the episode. And they can also follow along on Blue Sky to see how fascinating and interesting your life as a scientist is. So they might get a different perspective. They might change their mind. Who knows?
yeah, maybe they get interested when they see that there's also a lot of international collaboration where they get a chance to travel and so on and so on.
Absolutely. I would be happy if it changes their mind. That would be awesome. So thank you very much, Chris. This was really great. And hopefully I can have you again on the podcast to talk about your research, the fermentation, the industrial scale fermentation, so on and so forth. But yeah, this was really fun for me. Thank you very much, Chris.
Yeah, thank you.
Thank you for listening. I hope you enjoyed this episode. To find out more about us, visit our website, thesciencetalk.com, and make sure to follow Under the Microscope podcast wherever you're listening. And if you're watching on YouTube, make sure to subscribe to the YouTube channel of The Science Talk. Thank you for listening, and we will see you in the next one.